So next part is biological database. There is a note on different biological database. The first important biological database which one studies is the NCBI database. Apart from NCBI database, you have the XPACI database, you have the EMBL database, Swiss Prot database. There are several databases, biological databases. So let us check these databases one by one. Biological database. So basically, biological databases, what are biological databases? They are libraries of biological sciences collected from scientific experiments, published literature, high throughput experiment technology, and computational analysis. So they contain information from research areas including genomics, proteomics, metabolomics, microarray, expression, and phylogenetics. So, and the information contained in the biological database include gene function, structure, localization, both cellular and chromosomal, clinical effects of mutation, as well as similarities of biological sequences and structures. So any type of biological data which you find, they are essentially kept in particular servers and they are known as database. All these particular aspects, they belong to the biological database region. So the most important biological databases, let's talk about the first one, NCBI. It is the National Center for Biotechnology Information, NCBI. This NCBI database, it is part of the United States National Library of Medicine, which is a branch of the National Institute of Health of United States of America. So the NCBI is located in Maryland, Bethesda in Maryland, and was founded in 1988 through legislation sponsored by the US Congressman Claude Pepper. So this is a database relevant to biotechnology and biomedicine, and it is an important resource for bioinformatic tools and services. Major databases include GeneBank for DNA sequences, PubMed for literature. So if you want to have DNA sequence, protein sequences, you go to GeneBank. And now if you want to have all publications, you want to see bibliographic, you go to PubMed. This is all inside this NCBI database. Other database includes the epigenomics database. Entree, that means Entree is there. That's also for searching. And this is directed by David Lipman. And who was also one of the original authors of BLAST. BLAST is something you will understand very fast. This is something which you do very often. So let us go to the NCBI database and see what is there. ncbi.nlm.nih.gov. So this is the National Library of Medicine, National Center for Biotechnology Information. So this is the NCBI homepage. You can see there are so many resources. You have chemicals and bioassays resources, DNA and software resources, DNA, RNA resources, domains and structures you can predict from here, genes and their expression you can find out, genetics, medicines, genomes and maps, homology, literature, protein, sequence analysis, taxonomy. There are different trainings and tutorials also present, as well as variations. So people, they submit or deposit data or manuscript into the NCBI database, and they transfer the NCBI data to, then you can download it also. If you people submit it and you need that data, you can download it from the NCBI database. It is an open source, free for all. You can develop APIs, tools. You can analyze data. Then there are a lot of research things. You have NCBI news and news blog also is there. But in all the databases, most important database, let us see, is nucleotide, where you can get DNA sequences. Say, I want to have the DNA sequence of E. coli 5S ribosomal RNA. I type E. coli 5S ribosomal RNA. I press enter and I get all the sequences. Here, see this particular strain, Sakai strain of E. coli, I get the 5S ribosomal RNA complete sequence. I go inside this sequence. So you have all the information about this sequence. E. coli, you have the locus. This is this number you have to, this is the locus in the genome. This is 120 base pair. This is the ribosomal RNA linear. Then this is the accession number. You need this accession number to get the particular sequence. And author's reference, from which reference you can get the journal where you can find it was given first, name of the authors, the title, name of the publication. You get all data from about this. Apart from that, this is the particular sequence of the ribosomal RNA. Normally, this type of RNA is not very useful. This type of sequence, DNA sequence, is you need it in a FASTA format. So this comes as a FASTA format. This is the 5S ribosomal RNA of E. coli. Now, if you want to analyze this sequence, you want to see in the database of all bacteria, you want to see, you want to find out if there are any similar sequences. So you do a blast throughout the database. Now blast is taking place. Here you can paste this accession number, or you can paste the sequence in FASTA format. And you can have aligned 2 or more sequences. But we are not aligning, we are aligning one sequence. And so what you do is, you do blast. Throughout the database, it is trying to find out. Now let's see what error have I done over here. So let us take the sequence. So I'm taking this particular sequence, copy, and then I go to the home page once more. You find resource for blast. You are blasting the nucleotide. So you do the nucleotide blast. You paste the sequence here. E. coli 5S RNA sequence. You paste it over here. And you do a blast. Now it is working throughout the genome. It's finding the database. And you get your results. What results do you get? You get all E. coli, E. coli, E. coli, E. coli. So you get all throughout the database. You're getting similar sequences throughout. Clear? So basically, you can understand what this particular sequence is. So this is the next part. Is it clear so far? Yes, no?
Yes, sir.
Okay. The next part is that this can also be used for searching literature. So biological use searching literature. I go to the main page. Now I go to PubMed. Here, PubMed. Here I want to find a journal. Let's see my publication. I'm sort of having a narcissistic feeling about showing myself. So let's see if any publications are there about Arnab Ganguly. Many papers are present for Arnab Ganguly. You search the database. There is A Ganguly here. This is one of my paper which has come. You can find. Here's another of my paper. So you can see that you can find lots of papers of mine. They are coming up over here.
Sir, are you presenting the screen right now?
I am not presenting the screen right now. Thank you for reminding me.
Okay, sir. I was not understood.
So I'm presenting the screen right now. I'm going back. So like I said, I'm feeling narcissistic. So I want to show that what I have done to science. I want to find out publication by a person known Arnab Ganguly. So I go to PubMed, and there I type my name, Arnab Ganguly, and I give search. So you can see that all my publications come out over here. All A Ganguly is coming over here. Now I want to see what publications Arnab Ganguly has done on heat stress transcription factors. I give a plus HSF. See, these are the publications which I have done on heat stress transcription factors. These are the papers for heat stress transcription factors. Yeah, so like this, you can search for papers. Apart from that, you can find out books. Let's see, books by anybody. Give a name of Gilbert, say. See, Developmental Biology, 6th edition, Gilbert. Axis formation, you have all the chapters coming. So, this is a part you can have the whole book or details on Gilbert's developmental biology book. Okay, so this is the NCBI database. In the next class, today, since there is very low number of students, I don't want to do too much of the details. So I will stop here. Next day, we will check another database, that is XPASI. And if more students come, then we will do EMBL also. Okay?
Okay,
Any,
sir.
any questions?
No, sir.
Okay. Then thank you very much.
Thank you, sir.